Okay. That's my title, and I'll explain the hope bit as we go along. So I'm going to begin with a little bit of a personal indulgence. I hope you'll forgive me. The project I'm going to present today is close to my heart. You could think of it as an honors project that got out of control. So my work in Jamaica has basically been a career-long activity. I began it in the 1970s. So the analogy I wanted to make with this slide is if you're a first-year PhD student, and there's probably about 40 of you in the audience, and you've just got your first field season under your belt, then imagine going back to your study site repeatedly until 2051, because that's basically the equivalent of what I've done. So in the process, I think I've learned quite a lot about how coral reefs function, and particularly about issues like resilience and regime shifts. So the research questions I'm interested in are, how do regime shifts occur, and how reversible are they? What's the role of herbivory is the issue I'll focus on today. How does coral species composition shift as we go from a coral-dominated state to a macroalgal-dominated state? Most of the literature, and certainly all of the large-scale literature, is about what happens to coral cover and macroalgal cover. And finally, at the end of the talk, I'll talk a little bit about what are the functional consequences of shifts in the species composition of corals. So the scale of the study today, I'll focus on three elements. Briefly, I'll talk about the sea urchin Diadema antillarum, which is the primary herbivore on most reefs in the Caribbean. So the spatial scale of this study, where I've looked at three elements: Diadema, cover by corals and macroalgae, and finally species composition of corals, is island-wide. Depending on how you measure the coastline, it's up to 800 kilometers in length. The temporal scale, I realized to my horror this morning, is actually 5 decades, from the 70s to the 20-teens. And I'll be talking at the species level from about halfway through the talk onwards. So the story of the die-off of Diadema and the consequences for the growth of macroalgae and the subsequent loss of corals is quite well known, so I'll just briefly skim over it. So the graph on the left shows the pre-die-off densities of Diadema across a depth range, and I want to make 2 points. One is Diadema used to have a very broad depth range, with something close to 10 Diadema per square meter, all the way down to something like 18 meters in depth. Today, there's been very, very limited recovery. Most of the sites around Jamaica and elsewhere in the Caribbean that used to have 10 Diadema per square meter still have none. And where it has recovered, often the densities still remain lower than 1 per square meter, which would be very low down on that y-axis. So let's start out by just having a quick look at what happened to total coral cover and macroalgal cover through time. So that picture on the right, I took at a place called Rio Bueno in 1978, and it shows the two dominant branching acroporas, Acropora palmata and Acropora cervicornis, which used to completely dominate this site and many sites in shallow water along the north coast of Jamaica. So the plot is a graph showing lots of data points from many locations of macroalgal cover versus coral cover, and the color coding shows a hot spot in red and orange along the bottom. And it says that typically coral cover ranged between about 35 and 65%, whereas macroalgal cover was often very close to zero. The highest macroalgal cover locations would have been deeper mesophotic reefs over that time period. I'm going to skip an intermediate time period and go to the most recent period from 1989 onwards, when things stabilized. And you can see there's been a radical shift in the contours of coral cover and macroalgal cover. So the hotspot since the late 80s up to pretty much the present day is less than 10% coral cover and about 75% macroalgal cover. I also want to point out those 6 outliers with the arrows pointing to them. Those are the hopeful spots, and those, not coincidentally, are the only locations in my data set where Diadema has partially recovered. So there's been an enormous transition, and there basically have been 2 pathways over the last 40 years or so. One I would characterize as a lock-in, where we now have very low coral cover and very high macroalgal cover and no Diadema. And the other is this new trajectory, where there's an emerging, highly altered species composition of corals, which I'll get to next. So I want to show you 2 examples. One would be one of those outliers at the bottom right, with the arrow pointing to it, and another would be a more typical reef, which I'll show first, which is near the red hotspot on the graph on the right. So the plot on the left is a little bit garish. The colors represent different species. So it's a plot of species level cover through time from 1977 up to 2013. The green dots are covered by macroalgae. 2 vertical arrows show 2 critical events in this timeline. The first one in 1980, between 80 and 81, was Hurricane Allen, which caused a big crash in coral cover. And the second one to the right in 1983 was the timing of the die-off of Diademas. And you can see a picture of some very sick, actually dead, Diadema on the right. This was a very spectacular event where literally tens of millions of Diadema died over a period of about 10 days. So the top 2 species on the left of that graph before Hurricane Allen were the 2 branching acropores, Acropora palmata and Acropora cervicornis. They were killed off by Hurricane Allen, and they haven't recovered since. And that's typical of virtually all of the locations that I've studied around Jamaica. Coral cover initially at this site was close to 60%. It was the photograph I showed you earlier. Coral cover today is just under 1%. There are no Diadema, and this place has become so windswept, so flattened, that I think it would probably be next to impossible for Diadema to be reintroduced or to resettle there. So I'm calling this a lock-in, where we now have a semi-permanent regime shift from coral dominance to macroalgal dominance. A more hopeful example is shown, before I get to the more hopeful example, this is on the right here, is just a multivariate analysis of that shift in species composition. I'll be showing you a couple of those. So this, in multidimensional space, is the species composition of the corals through time. So on the left of that graph, you can see there's a cluster of assemblages that are very similar in 1977, 78, 79, and 80. Then there's a jump in species composition caused by the loss of the acroporas to a new cluster in the early 1980s. And then over the next decade, we go through a decade-long transitory phase, which culminates in a new cluster, which is basically very little abundance of all the corals. So this is very much analogous to 2 basins of attraction, or a regime shift with a decade-long transition phase. And this is typical of the vast majority of the locations that I've measured this composition through time. Here's a more hopeful example where we, in the last recent period, are seeing a recovery. This is slightly deeper. This is at a depth of 10 meters. So again, it's species composition color-coded on the left through time, and the green dots are macroalgal cover. And you can see that macroalgal cover began to decline between 1990 and 93 with the beginnings of a partial recovery of Diadema. And in 2013, Diadema are now back up to about half of their former density, about 5 per square meter, and they've demolished the algae. So the picture on the right is actually looking pretty hopeful. Coral cover has gone from a low of 5% in 1993 back up to just over 30% cover. What's different, though, is the species composition. It's highly altered. So the brown color, the fourth one down on the graph on the left, which you probably can't see terribly clearly, is Acropora cervicornis, the staghorn coral. It's gone. The yellow one is Agaricia agricites, and the red one is Parietes asteroides. So those are now the two dominants, but they're only now approaching the former abundance that they had before the die-off of the Diadema. And this is the best case I can show you of all of the study sites that I've been examining for a long time. So if we pool all our sites and look at the species composition there, first I'll show you what's happening here. So again, we have a cluster, a stable assemblage prior to Hurricane Allen. You can think of that as being analogous to a basin of attraction. The system was bumped out of that basin of attraction by Hurricane Allen, and following the Diadema die-off in 1983, it went for a long walk before it stabilized for a long time at a very degraded system like the one I showed you earlier that is locked in. But what's different here is that with the recovery, partial recovery of Diadema, we now see a huge leap back to the left between 1993 and 2013, where there's been some recovery of coral cover, albeit with a very different mix of species. So we are now on uncharted territory in terms of the species composition of corals where this recovery is happening. But I want to emphasize that this is a small minority of locations where this kind of recovery is taking place. So if we pool all of the sites and do a multivariate analysis, in the early period it looks like that. So there are 3 clusters or hotspots. The one on the bottom is shallow, the one at the top is mesophotic reefs, and the one in the middle is mid-depth range locations. So because of the structure of the data, which is depth related, we can see the unique species composition that used to occur at different depths. If we do that same sort of analysis today, it looks like this. So regardless of the depth, all of these sites have converged on a low coral cover, low abundance of every coral species hotspot, which is shown on the right. And for the sites that I have species compositional data for, there are only 4 which have left that high concentration of low abundances indicated by those arrows, and those are the 4 locations where Diadema is recovering. Where they'll end up is anyone's guess, but we're on uncharted territory in terms of this new trajectory. And if we superimpose those graphs, you can see that there's actually no overlap in current species composition from the historic composition, which was classically described by Garreau and others when they described the Acropora palmata zone, the Acropora cervicornis zone. Those are gone, and other species compositions at various depths. I'll just finish up by talking about some of the functional consequences of this radical shift in species composition. I want to make one point first, and this is the vectors for that multivariate analysis. So macroalgae are to the right, so the shift in the hotspots from the left to the right represents in part the huge increase in macroalgae and the loss of Diadema. The important point I want to make here, though, is that all the other vectors are for the abundance of individual coral species, and they all point that way. There aren't any coral vectors pointing this way, and that means that today the abundance of all coral species is depleted across all these sites. So at a seascape scale, no coral today in Jamaica is as abundant as it used to be. And I'll just reinforce that here with this example. That photograph there was taken in about 1977, and it shows the three dominant species, which are also shown in the bar graph over there. So number one was the branching coral, Acropora cervicornis. Number two is the massive Orbicella annularis, and number three is Acropora palmata. Those are all incredibly depleted today. So all of the species represented in the bar graph used to have an abundance at the species level of at least 1%, and some of them were much more abundant than that. So on average, 9% for cervicornis, 7% for annularis. Those are the kinds of numbers that most modern coral reef ecologists working in the Caribbean would be quite happy to have for all the species combined. But there's been a huge shifting baseline in terms of what the species composition was and what the species level abundances were. So what's happened to that through time, I'll show you in a second. So using a trait-based approach, one of my students, Mike McWilliam, has been looking at three-dimensional functional space, if you like, for this assemblage and how that's changed through time during a regime shift. And so this is the analysis that Mike has done. So the colors here in the circles correspond to the colors in the bar graph. The size of the circles corresponds to the height of the bar. And so this is a multi-dimensional analysis where the center of each circle represents the function determined by traits like whether it's a brooder or a spawner, its morphology, its growth rate, and so on. So on the top right, there's a cluster of species that are all weedy brooder species. The yellow one is Agaricia agricides. In the bottom left, there's two circles that strongly overlap. Those are the two highly three-dimensional, fast-growing Acropora species. The purplish large dot on the left above the two Acroporas is Orbicella. So what happens to that functional space through time? The bars on the left show the contemporary species level cover, and the y-axis has changed in scale, so you'll have to trust me that the yellow bar for Agaricia agricides in 2013 is just approaching its former abundance, but still not greater. And the second highest bar in red is Parietes astrioides. The main point I want to make here is the functional analysis, the circles on the right and the bottom for 2013, all of those circles are depleted. So in terms of the functional roles of this assemblage, not only have we lost lots of abundant species, but every function is depleted. So things like calcification, photosynthesis, habitat provision are also all depleted. So in conclusion, I hope that wasn't too depressing. I think this study, which has lasted my entire career, shows that regime shifts and reversals in the coral reef realm unfold slowly, generally, and that herbivory plays a huge role. There have been lots of bleaching events and disease epidemics, particularly in the last decade among the corals. But what these results show is that herbivory trumps them, in that if Diadema comes back, we do get a recovery, at least of coral cover, and at least of some of the more resilient species. A second result is that shifts of composition occurred during this study as coral cover went down, as coral cover went up, and even as coral cover stayed the same. And I guess my point is that coral cover doesn't tell you a hell of a lot about what's really going on in terms of the functional dynamics that's unfolding during a regime shift. And finally, major functional consequences are unfolding as these shifts in composition. And even when coral cover is increasing, it doesn't necessarily mean that these functions are being restored. I think the scope here for people to take on board these results for management, and in particular, I think there's a lot of promise in moving Diadema around. They are recovering at a small minority of places and are starting to reach densities that are probably now getting too high. And so there's scope to spread the Diadema around, get rid of the algae so that coral recruitment can once more resume. Thanks very much.